Hello, my name is Aditya Sripathi, and today is the 4th of October 2019. I am making this video for my Unit 2 case connections. I've also made a PowerPoint presentation with regards to case 2 and case 4 of my Unit 2 case connections assessment involving some pictures and bullet points of what I'm going to be talking about, and I reference a slide at the end that I've also attached to the script. We're going to get into the PowerPoint presentation now, basically discussing the solutions for case 2 as well as case 4 of my Unit 2 case connections. So hello again, this is the PowerPoint presentation that I've made with regards to my Unit 2 case connections assessment. I have essentially chosen case number 2 and case number 4 for this video presentation. I will first start off by discussing case number 2, describing it, and then we'll get into the solutions component of it. After which we'll get into case number 4, describe that, and get into the solutions component of that as well. Now, this over here is my description for case number 2. As we can see here, case number 2 provides us with an image of a brain. We have an MRI scan over here as well as a PET scan over here. In the MRI scan, near the middle left region of it, we see an extra white region over here, as shown by the cursor. The MRI scan essentially shows a large glioblastoma with necrosis in some parts of the tissues. And the PET scan, as we see over here, is responsible for showing us a high absorption of D-glucose as indicated by the fluorescence, the bright orange fluorescence. This is basically carried out by having fluorescent proteins incorporated into glucose, and basically when it glows, it suggests that there is high levels of D-glucose present. This shows that the cancer cells in the tumor have also absorbed more glucose than the surrounding tissue, and for such a thing to occur, certain kinds of cellular modifications would have to happen so as to allow for increased glucose absorption. Now, the first question that's asked in case 2 is for me to essentially look into how glucose uptake is regulated in the types of tissue mentioned here in the table. So in the liver, the glucose transporter that is used is GLUT2, and it is not dependent on insulin. This is very similar to the brain, where it is not dependent on insulin, and it is also glucose uptake is also carried out by two different types of transporters, that is GLUT1 as well as GLUT3. Now the tissue type for skeletal muscle involves a glucose transporter known as GLUT4, and it is insulin dependent. And the same goes for adipose tissue, where the glucose transporter is GLUT4 and is insulin dependent as well. The dependency of muscles and adipose tissue on insulin will be described in the next slide. Now the GLUT4 transporters are mainly found in the adipose tissue and skeletal muscle. When insulin is released by the pancreas, especially during digestion, it binds to certain kinds of receptors on the cell membrane over here called receptor tyrosine kinase. The receptor tyrosine kinase then dimerize and trans-autophosphorylate each other's tyrosine residues on their intracellular domain. This then acts as a docking station, allowing for a particular signal called as IRS to bind, which is known as your insulin receptor substrate, and ultimately is going to be responsible for carrying out exocytosis by activating certain kinases, allowing for the exocytosis of GLUT4 transporters to the cell membrane, as seen over here. Now, an increased exocytosis of GLUT4 transporters to the cell membrane allows for more receptors to be present, thus leading to increased glucose uptake. In the absence of insulin, however, endocytosis of this GLUT transporters would take place, thereby leading to the absence of any GLUT transporters, and as a result, no glucose can be taken up by your cell type, in this case either your adipose tissue or by your skeletal muscle. Now, glucose uptake in normal tissues is definitely lesser than in tumor cells, and this answers question 2 of case 2, which essentially asks us if glucose uptake in normal tissues as compared to tumor cells is different, and what would the advantages of this be. So glucose uptake in normal tissue is lesser than in tumor cells. This is shown in the PET scan as indicated by the fluorescence of D-glucose in the cancer tissue. It would also be advantageous as increased glucose uptake leads to increased glycolysis, which provides more energy. The energy can then be utilized to support cellular processes, allowing the tumor to proliferate. Now, the modifications that might have occurred in the cancer cells to alter glucose uptake answers question 3 of our case. One of the modifications that we strongly believe, or I strongly believe, is the increased exocytosis of GLUT1-3 receptors to the cell membrane. An increased number of receptors definitely leads to an increased glucose uptake, and it can ultimately lead to the establishment of a positive feedback mechanism. Now, the positive feedback mechanism is very simple. An increased glucose uptake leads to increased energy in the cell, and that increased energy leads to increased protein synthesis. An increased protein synthesis leads to protein synthesis such as GLUT1 and 3 transporters, which lead to further glucose uptake, and the cycle reinforces itself. The next case we're going to be looking into is case number 4. It looks into a 2-month-old female baby that shows multiple symptoms such as exhaustion, as the baby sleeps more than 2 hours between various feedings. The baby also shows hypoglycemia, and the baby also shows hepatomegaly, which is essentially an enlarged liver. The baby is also only fed breast milk. As a result, the only source of sugar that enters the baby is galactose and glucose, which is essentially lactose. Epinephrine was also administered by the doctor, but no changes observed in the glucose level, and the blood glucose levels do increase immediately after breastfeeding. However, they do not remain at that level for long periods of time, especially the fact that it's not maintained during the fasting stage. Now, the solution to this. The question asked to us by case number 4 is for us to diagnose what the patient is suffering from, and the diagnosis is galactosemia. The patient over here in the case is provided only breast milk, which is essentially lactose, and we know that lactose is a disaccharide that is composed of both glucose as well as galactose. The galactose, as we see over here by my cursor, is then phosphorylated with the help of galactokinase to form galactose 1-phosphate. Galactose 1-phosphate then is acted upon by an enzyme known as GALT, or galactose 1-phosphatidyletransporase, as seen in this orange box, to eventually convert it into UDP-GLC. The UDP-GLC can then be acted upon by glycogen synthesis to form glycogen, and ultimately the chain takes you to glucose. Now, when GALT is defective, which is the enzyme responsible for converting it from galactose 1-phosphate to UDP-GLC, it prevents the conversion of all of the galactose 1-phosphate to UDP-GLC, and therefore all the galactose that's consumed by the baby remains as galactose 1-phosphate. The next thing is epinephrine administration had no effect on blood glucose levels. This is due to the impaired activity of GALT. Since GALT does not work, galactose remains stuck as galactose 1-phosphate and does not produce glucose. This process of phosphorylating galactose uses up most of the phosphate form from ATP, and hence prevents glycogen phosphorylase from being phosphorylated. This prevents glycogen phosphorylase from being active, as it requires phosphate for its activation, and hence it no longer breaks down glycogen when epinephrine is administered. The patient also suffers from hypoglycemia due to the fact that she also suffers from galactosemia. This is because when you suffer from galactosemia, and with lactose as the only source of nutrition, glucose is the only source of energy or sugar that the patient receives, and hence immediately gets utilized post-feeding. It is also due to this reason that blood glucose levels are high right after breastfeeding, but drop down eventually, as galactose is no longer a source of fuel, as it is stuck in the galactose 1-phosphate form in the liver. Galactose, on the other hand, therefore does not contribute to blood glucose levels. Now, there are multiple other options provided to us in this case. We know that it is not hereditary fructose intolerance. This is because there's no fructose provided to the patient, although hypoglycemia is a common symptom, and therefore we cannot consider hereditary fructose intolerance as a possible scenario for the patient. It is not glycogen storage disease either, as epinephrine would have definitely led to a decrease in the glycogen content, causing it to be broken down into glucose, allowing for blood sugar levels to be stabilized. It is also not glucose 6-phosphate dehydrogenase deficiency. This enzyme is known to convert glucose 6-phosphate into 6-phosphoglucolactone. Deficiency in this enzyme will lead to increased glucose 6-phosphate, which could then be converted to either glycogen or glucose and could have rectified blood glucose levels. But since the glucose levels were not rectified, we know that this deficiency is not a possibility either. Therefore, the solution for case 4 is galactosemia due to the various aforementioned data. These are the references that were utilized during this presentation. I've also attached them in the script. I hope you have a good day. Thank you.